Okay, hi everybody. My name is Jim Gell. I am the host of this here, Grit City Think and Drink. I'm a professor at the University of Washington, Tacoma. And I should give a shout out, which I keep forgetting, to my employer, the School of Interdisciplinary Arts and Sciences, who helped sponsor this event. We have had sponsorship from the bar that we usually do this in, but since COVID is still going, we are waiting to go back in person until sometime undisclosed at this point. So we'll figure out where we're going to be when we get to go and when we get to go back in person. So hopefully that's sometime soon. For those of you who don't know, this happens the second Tuesday of every month. So our speaker tonight is Dr. Kristen Hultgren. She's going to give us a talk here in just a second after I introduce her. But just to give a shout out for our next talk, Dr. Jessica Ray from UW Seattle in civil engineering is going to talk about how you make materials to actually take out specific contaminants from stormwater. She's doing all kinds of cool things to kind of invent new materials to take out specific problem chemicals rather than using our general approach we've done before. So I hope you'll tune in October to check out Jessica Ray. And then we have the rest of 2021 scheduled to come back in November and December and see what's up. We are looking for ideas for 2022. So if you have them, you can chat them to me anytime in here. If you have speaker ideas or general ideas for types of talks you'd like to see, go ahead and chat those to me and I will take note. So the way this works, for those of you who haven't been here before, I'll introduce the speaker. She'll give you an amazing talk. And then she'll take a short break while I give away, oh, I didn't grab them out, a pair of Grit City Think and Drink socks. So we go through the wardrobe. We've had hats, we've had shirts, and now we're on to socks. So make sure you get your limited edition collector item socks so that we can all look the same and you can show off your pride. So we'll be giving those away after her talk. We'll ask a couple trivia questions for you to chime in on. So you're going to use the chat for that. For those of you who haven't done this at this point, you're probably living under a rock, but just go to the top, to the bottom of your screen, find the chat thing, and you can chat directly by typing into there. All right, so and then after that we'll do a question and answer. So you chat your questions to me, I will feed them to Kristen, and you can play Stump the Speaker at that point and see what Kristen knows. So without further ado, I'm going to stop sharing my screen. And I'm going to introduce our speaker. Dr. Kristen Hultgren is an associate professor of biology at Seattle University, where she has worked since 2012. Her research spans several topics, including marine biodiversity, shrimp taxonomy, and evolution and ecology of snapping shrimp. She earned her, I just feel like we need to do that. She earned her BA in biology at Brown University, my alma mater as well, and her PhD in ecology at the University of California at Davis. Without further ado, Dr. Hultgren, it's all yours.
Hi, thanks. All right, let me do the screen share and then get this started. Hold on a sec. And then minimize. All right, thanks a lot. So when I talk about marine biodiversity, I think the first thing that comes to a lot of people's mind is these really archetypically diverse places like tropical coral reefs. So places like the Great Barrier Reef in Australia are really quite diverse. There's something like 1,500 species of fishes, maybe 300 or 400 species of corals. But where I think a lot of the biodiversity lies that we don't really see, and thus we don't really know much about, is what is called the cryptic invertebrates. And those are the invertebrates that live inside, if you were to kind of bust open a dead piece of coral or kind of look in the spaces between rocks, you can find often something like 200 different species in a single coral head. And those are going to be lots of invertebrates, so these animals without backbones, things like snapping shrimp, my favorite. They're actually a big component of the diversity. And then also cool things like crabs, these assorted worms and nudibranchs, and lots of other really cool critters. So if you want to study diversity from kind of a sheer numbers perspective, you study invertebrates. But you really don't have to travel to something like a tropical coral reef to study biodiversity in the ocean. So within Puget Sound and the Greater Salish Sea, where I'll be kind of talking about today, there's really hundreds of different species living in all these diverse habitats. And today I'll talk about crustaceans, which are kind of my area of expertise. And these include things like crabs and shrimp, which you see here, hermit crabs, but also things like amphipods and isopods up here. I think a lot of people would be really surprised to know, and I hope to kind of convince you today, that Puget Sound is really exceptionally diverse for a temperate habitat. And one of the things I'll talk about today is kind of how we can measure biodiversity and how it kind of stacks up to other temperate and tropical habitats around the world. But first, there's a couple challenges in studying marine biodiversity. First of all, it's actually really hard to identify a lot of these different species. For some of the more obscure taxa, you really have to be a taxonomic expert. And many species look alike, and they're really difficult to figure out which actual species they are. And then oftentimes within a single species, there can be a lot of color variation. So one species may look really different. So these are all species, this is all one species from the Puget Sound with these just like amazing color morphs, but it's the same. And then you can also have two very similar looking species, or two very similar shrimp that actually turn out to be different species, even though to most people, they look pretty much the same. So that's one challenge. And the second challenge is that another kind of surprising fact is that we still don't know a lot about the diversity in the ocean. So there's still a lot of undescribed marine species that are kind of new to marine science or new to science. So, but like in the estimates, there's something like between 30 or 90% of marine species are still not formally described. And the fun part about it is that scientists get to describe and name the new species they find. So myself and a couple of collaborators in Panama, we found this really cool shrimp from kind of Pacific side of Panama, and it has this amazingly pink claw. So we named it Synalpheus pinkfloydii. This is like the most press I've ever gotten in like my whole life, is naming a shrimp after Pink Floyd. So that's the kind of fun part. So how do you overcome these challenges? So one really simple way that's becoming kind of increasingly common and cheap is to use what's called genetic barcoding to describe diversity and to kind of find potentially new species. So there's lots of fancy infographics on the web, but I really like this comic book, or this little comic of it. So if you have an unknown organism, imagine this is a shrimp instead of a bug. You don't know what it is. Maybe you're not an expert in that group. You can pull off something like a leg. You can extract the DNA and amplify a really common gene, that is, make a whole bunch of different copies of a really common gene that a whole bunch of other people have sequenced. You sequence that gene, and then you can look up your sequence inside a big database of barcodes. So one that you might have heard of is something called GenBank. All scientists, when they typically publish their data, publish their sequences on GenBank. And then if you don't know what this critter is, maybe you'll get a match, something like a 99 or 100% match on GenBank, and boom, you have a species name. So for invertebrates, the most common gene that we sequence is called cytochrome oxidase 1, or CO1. And I won't talk about the methods today, but we can also use CO1 barcoding to kind of discover potentially new species. So basically you'll sequence two things that look the same, but it turns out their sequences are different enough such that we think that they're, or kind of like tentatively call them two different species. So that's one really cool solution, and you can do it relatively quickly and cheaply these days. Another challenge is that it can be often difficult to kind of sample standardized biodiversity habitats underwater without actually destroying that habitat. So when I said you can find something like 200 different species in a dead coral head, you actually have to wrench that coral head off the reef and kind of bust it open with hammers, which everybody does feel a little bit guilty about. And many times when marine ecologists have problems, we solve them with marine ecology arts and crafts. So these are the arts and crafts that I'll talk about today. These are little structures called ARMS, or autonomous reef monitoring structures. It's basically just a bunch of plates that you kind of sand and stack together with these little spacers together to make these little apartment houses. And then you actually bolt these arms. They're about this big, if you can see my video, a couple, like maybe a foot on each side or a little bit less. You bolt them underwater, maybe somewhere between 15 to 30 foot depth. And then you come back after a couple years and you can just detach these whole arms and bring them back to shore. And then you can get a really standardized view of kind of biodiversity per square unit. And this is what they look like after a couple years. So these are some arms. This is actually a really cool picture from some arms that somebody else set out in the Red Sea area. So there's a little sea cucumber there, and this like squid is about to do something interesting. So because these are kind of cool standardized devices, you can use them to study biodiversity around the world, not only in reefs, but in places like Puget Sound. So today, I'll talk about studying the crustacean component of diversity in the Puget Sound and using ARMS to kind of get a standardized measure and compare it with biodiversity of crustaceans around the world. And then I'll talk about another local study that I've done with some undergraduates, where we looked at what factors just in the local kind of Puget Sound, Salish Sea area, what factors are correlated with crustacean biodiversity. So for this first question, how does our biodiversity stack up against everywhere around the world? From about 2016 to 2018, ARMS were deployed about 5 sites around the Salish Sea, all the way from down south in Nisqually, up north to Friday Harbor and Shannon Point, and then west to Neah Bay. So everything here but the Friday Harbor ones were set out by NOAA, and Gustav Pauli from the Florida Museum, one of my collaborators, he set out the ones with his invertebrate zoology class up at Friday Harbor. So we'll see a little video of some of these in a little bit. So I was lucky enough to help with the recovery of the Nisqually arms on kind of a rainy day in the spring. These are some pictures of bringing them up from the dive boat. Again, you'll see a much better video of this. Kind of disassembling them and then sieving them through. The invertebrates I'm going to talk about today are put through a little like mesh sieve. So we're looking at things that are kind of over about 2 millimeters in size. All right, so we're going to do a video. Here we go. Yes. And this video actually is really cool because it features one of my collaborators, Gary Winans from NOAA.
So understanding biodiversity is important. Biodiversity is what holds an ecosystem together, which gives it resilience during change. This is a project we're trying to catalog biodiversity in Puget Sound. We're trying to look at the small animals that are overlooked and understudied in Puget Sound. So about 2.5 years ago, scuba divers went in and put in these structures called ARMS. They're actually a series of plates, plastic plates with openings between them. Things came and went over 2.5 years. Things attached, things swam through. They're like little invertebrate hotels. So today we brought them up. 2 divers went down. They drop a cage on top of the arms, invert it and bring it up. So we've captured what was living in there. They're carried in totes, so everything's still alive. So when we bring them into the laboratory, we unbolt the thing, we take the structures apart, so all 9 plates are swished, and we get all the mobile animals into a big tote. We sieve them out. Anything bigger than 2 millimeters is taken into the lab. We get to pick through and try to identify what's been down there. There's crabs, there's shrimp, We see sea stars, sea urchins. We saw 3 flatworms that just ooze their way around on the plates. One of the coolest inhabitants of this thing that we've seen at every location is this pygmy Eula shrimp. It's a ½ inch at most. And we found females are just totally full of eggs, and we got 150 of them in one spot. Yet, who knows about that? I didn't know about that. I'm excited to be able to photograph those and put them in the category for Puget Sound. So it's a 2-step process. We identify all the species we can by eye that are greater than 2 millimeters. Everything else, then we extract DNA from the unknown things that we can't quite see. So by gathering information here, by doing the first inventory at this level in Puget Sound, it lays the foundation for future work. What is there now? What is there in 10 years? And I think by keeping an eye on this inventory, we're better able to look at models of ecosystem change and how is the ecosystem responding at all the different levels.
All right. There we go. Perfect. Yeah, that's a great video. That really says it very clearly. I love all the videos. So the arms were deployed. We kind of picked them all up and scraped them all out and got all the little invertebrates in jars. And what I really chose to focus on is the crustaceans, because that's kind of where my taxonomic expertise is. So I had a research class in marine biodiversity in spring of 2019, and we barcoded a pretty large sample of all of those. So these folks really helped with that part. And then I also had a lot of jars to look through, and an opportunity in spring of 2020 to stay home with my microscope and just look at a lot of shrimp and crabs. So here are some of the photos of all of the cool critters that we saw. Out of those those 5 locations, about 3 arms at each location, we looked at about 2,500 individual crustaceans and about 39 different crustacean species. Again, there's a sample kind of shown here. And of them, we have what we think are at least 2 potentially new species. So that little Eulid shrimp that Gary was talking about in the video, that actually turns out to be 2 shrimp. When you look at populations from different parts of the sound that are very genetically distinct, it's really hard to tell them apart morphologically, but this is something we're going to look at later. So that was cool, just to get this really nice biodiversity inventory, and again, with lots of pretty pictures, which are going to be very useful in the future. And then because we use these arms, we're also able to kind of compare our estimates of biodiversity with other sites worldwide. So here's us in the Salish Sea. And we compare crustacean biodiversity in our samples with biodiversity in Virginia, Florida, the site up in the Gulf of Aqaba in kind of Jordan, and then Hawaii and Australia. And it turns out this is average number of species per arm. So it's kind of a good relative measure of biodiversity between these regions. It turns out that Puget Sound has a pretty high diversity in terms of crustaceans. So of course, the Great Barrier Reef in Australia is quite high, as you might expect, as is Hawaii. But we are pretty comparable with these, subtropical locations, Florida and Virginia, which is a nice surprise, and this kind of isolated region here up in the Red Sea. So that's kind of how we look compared to the rest of the world. But it also turns out that inside the kind of Puget Sound up to the Salish Sea up here, diversity is much higher in the northern sites. So we have a lot of regional variation as well. Again, here's Neah Bay and Friday Harbor, average diversity per arm. We only had 1 arm from Shannon Point because 2 of them kind of washed away. And diversity at these 3 northern sites, which are kind of close to the entrance to the Pacific Ocean, are much higher than these southern sites. So, and they're also much more similar in terms of like the crustacean community. They have a lot of similar species up here and down here. So this also kind of brings up the question of what local factors besides this kind of north-south trend, which I'll circle back to, might affect crustacean biodiversity. So this kind of is a nice time to talk about another local study that I worked on at the same time at the ARM study. And this really focuses on intertidal crustaceans. So marine intertidal communities, this is kind of intertidal out by Port Angeles on the Olympic Peninsula. They've been a really cool system in community ecology for looking at like what kind of factors affect how many species you have at the site. And this is a good time to kind of generally talk about how that there's different ways to measure biodiversity. So you can measure just the number of different species. That's really what I talked about in the first part of my talk with the arms. Or you can kind of calculate a number that's technically called species diversity, and that takes into account not only the number of different species, that's what we call richness, but the relative abundance. So if you had 10 different species, but most of your sample was made up of just like one really common crab, that site would have a lower diversity than a site which also had 10 species, but like equal amounts of each species. But recently there's been all these other new kind of ways to measure biodiversity. So I'm an evolutionary biologist. And you can also measure what you call phylogenetic diversity. And this is just, it's basically just mapping an evolutionary tree onto the different species you see in a community. So a site or a location with a higher phylogenetic diversity just has more species that are more kind of distantly related in this evolutionary tree. So if a species had this barnacle up here and this crab down here, that would have a higher phylogenetic diversity than a site that just had these two really closely related crabs here. And then finally, functional diversity, which makes a lot of intuitive sense. This is just a way to measure, using the number of species in a community, how diverse those species are in terms of all the kind of functions that they perform. So one example in crustaceans is that they have a lot of different kind of feeding functions, and that kind of makes them play different roles in a community. Everything from filter feeders to like these little vegetarian isopods that are mostly eating seaweeds, to kind of higher level predators, and then these omnivores and scavengers. So I'm going to study these three types of diversity just in terms of intertidal crustaceans. So again, this was kind of a regional survey, same map, different dots at about 11 different sites. And I did this work all with different components of this work with different undergraduates. And we basically looked at the invertebrate communities that were under boulders. So we flipped over boulders and we looked at the critters that were kind of underneath the actual boulder itself, and then on the surface underneath that boulder. And anyone who's really been out intertidling knows that most of the interesting stuff you find is under boulders. And then we measured a bunch of kind of environmental factors at each of these sites. So salinity, how salty the water is, boulder size. Some of the sites had really big boulders, some of them had big and small, and some of them had small. The kind of ruggedness of the shoreline, so how, if the shoreline was really flat, it would be less rugged than if it had little boulders and lots of crevices. And then we used photos to measure kind of how much of that the shoreline was covered with seaweed, otherwise known as algae, and how many different types of seaweed there were. So we'll just call that algal species richness. And then we were also interested in, trying to get some measure of urbanization. So the sites around Seattle are going to be a lot more urban than the sites around here. So we used GIS to measure a very simple measure of urbanization. It's just the percentage of paved surfaces within about 1 kilometer of our sites. So again, all these sites around LA Bay Marina, it's going to be a lot of paved, impervious surfaces. It's very urban, whereas up here and down here, kind of more natural terrain around our sites. So this student, Logan, helped me with all the field work. My student Kalia helped me really work on the phylogenetic tree, or the evolutionary tree of all these different species. And we use that as a kind of tool to figure out phylogenetic diversity. So that's like one number, how evolutionary complex were all the crustaceans at this site. And then Amelia thought she was going to do field work, but she helped me in spring of 2020. And she sat and figured out, helped me figure out all the functional diversity values. So we took all the little crustaceans that we found, we figured out what kind of functional traits they were, and then you feed all those numbers in and you get kind of a functional diversity number for each site. And then we kind of saw how these diversity indexes were related to all those environmental factors that we measured. And it turns out that algal species richness, or just how many different types of seaweed were there, that was the most important factor. So that was really positively correlated with all of the different ways to measure diversity. And this little picture here is just how we measured it. Here's a picture of our little measuring thing, and there's about at least 4 or 5 different types of algae or seaweed that you can see in here, right? And so phylogenetic diversity increases with the number of algae species here. And so does regular old species diversity. That also goes up. Functional diversity also increases with the number of different algae species. But functional diversity also was kind of correlated with, positively correlated with larger boulders. So those two variables, the number of different algae species, or algal richness and boulder diameter, those were really the most important factors, and none of the other kind of environmental factors really correlated a lot with biodiversity. And then we found this interesting trend. This isn't something we necessarily looked at in the study, but when you look at the data later, you can find that the diversity of different crustacean species, again, here's just the number of different species, is much higher in the northern sites than in the southern sites. So this is our study, but it's really interesting when you start looking at other studies of the Puget Sound. This is a study done in 2005, and this is shoreline. It's primarily shoreline invertebrates. So they surveyed a bunch of these sites from kind of south to north here. And they also found this south to north gradient where the sites down here had a lower number of different invertebrates than these northern sites. And then when you look a little bit further, this is a study, and there have been many more kind of related to this, of fish and jellyfish diversity. So this is a little different. Here's all the northern sites. So these are sites up in the Rosario and Whidbey basins here. And they have a higher number of different fish and jellyfish species than these southern sites here. And these are just at different times of the year. So really any different time of year, the diversity of fish and jellyfish is higher up here than down there. And of course, that also matches what we see in our ARM studies, right? Those little subtidal apartment houses that we stuck around here. We had much higher diversity in these northern sites compared to the southern sites. So there's something going on. It's not entirely clear. It'd be something that would be great to look at with some other studies. And what we think is going on is that it's really, a lot of this is really just, it's not necessarily north to south. It's really influenced by kind of how close these sites are to the mouth of the Pacific Ocean, right? So lots of the invertebrates that we're studying, they have these little tiny babies that they, when their eggs hatch, they hatch into these things called larvae, which swim in the water column. It's basically what we call, what you might think of as plankton. And so they can spend, weeks to months in the water column, and so like in the open ocean. And so these sites here might just have more larvae coming in from the Pacific Ocean, right? From other sites all the way up and down the coast. And it might be harder for those larvae to get all the way down to the South Sound. You would really need to look at this with some other studies. And then lots of animals just kind of prefer these more oceanic habitats, maybe areas with higher wave stress and stronger currents, right? They're animals that are seen a lot on the outer coast. And so they might be seen at sites like Neah Bay, maybe Freddie Harbor, but they're not going to be seen in these really calm areas down here. So, as I finish up, I actually think it's useful just to take a step back and look at the kind of broader perspective about why do we care about biodiversity? So why does diversity matter? And there's been like, ecologists are very curious about this. So is there some way to figure out how a higher number of species can contribute to ecosystem function? That is, positive things that we get out of ecosystems. And there have actually been a lot of studies on this, both experimental and kind of surveys. And it turns out one of the kind of classic patterns that you see is that more diverse communities are more productive. So again, here, as you increase the number of different species you have, the overall biomass of like living stuff in the ecosystem, what we might call productivity, increases, right? So if you want to maximize the amount of living biomass of living stuff in your community, you try to have a really diverse community. And that's been seen in lots of different studies, both experimental studies where they like made little communities of 1, 2, or 4 species, and then where they go out and actually survey species. So this has been found with things like seagrass beds, wetlands, coral reef fishes, and intertidal seaweeds. In some cases, more diverse communities can repel invasive or non-native species. So the kind of classic studies on this was on something called fouling communities. So if you go to Elliott Bay Marina or any kind of dock by the ocean, and you look on the side of boats, or maybe on the columns or the pilings, you find a lot of stuff growing on them, right? And those often have a lot of invasive species, so species that aren't native to our community. But it turns out the more diverse these fouling communities are, the fewer invasive species they have in these experimental studies. A little bit of data on this for intertidal seaweeds too. So if repelling invasive species is something that we want to achieve in our ecosystems, it's good to have diverse, healthy ecosystems. And I think this is a slightly different class of studies, but this is like some of the most exciting work that I've seen, is that more diverse communities seem to be more resilient to change, especially things like the big global changes that are occurring and that are going to be occurring in the future. And so these studies are actually on genetic diversity. So in things like seagrass beds, there's actually seagrasses that live underwater. And if you have more genetically diverse seagrass beds, when these heat waves hit, they turn out to be more resilient to like these really like hot water events. And the same thing was seen in some kelp forests. So this is one species of kelp, but if you have a lot of genetic diversity within that kelp forest, after a big heat wave, this is some work done in Australia, you find kind of a community that has resisted change better than if you have not a very genetically diverse community. And that's kind of what you see in A. So I think this is really exciting, this idea that diversity, at least genetic diversity and maybe species diversity too, can kind of factor into resiliency. So I'm not going to summarize everything again because I think I covered it pretty well. I did want to kind of thank all my collaborators. So in the Arms Crustacean Project, Gary from NOAA was really the one who told me about this study and got me involved. So I'm really indebted to him. My students in my class that helped me barcode, Gustav, who did a lot of the identifications and put out the Friday Harbor arms, and then Matt Leray from the Smithsonian, who shared all the data from all the other sites around the world. And then I had these 3 excellent undergraduates, all graduated now, that worked on the intertidal crustacean study. So with that, I will take some questions then.
Awesome. Thank you very much, Kristen. That was wonderful. I have so many questions, but we'll hang on for just a second so we can give away some freebies before we do that. So we are going to, so everybody get their chat fingers ready. We're going to ask you 2 questions. And so your prize is a pair of, oh, look, weird background. Think and Drink socks. These are awesome collector's items, and Christmas is just around the corner, so you need to win these. So also I should ask before you put in your answers, if you win, I am not with you physically right now, so I need to know how to get the socks to you. So if you chat your mailing address to me, only I will see that. I will send you the socks, and you also need to let me know whether you are, what size foot you have, whether it's women's or men's, and I will guess appropriately because I think they're like unisex small versus medium/large. So I will get you a pair of socks that hopefully fits your feet as close as possible. So again, if you win, please send me your mailing address and your size socks. So we're gonna give away a couple pairs. So the first one is Kristen, or Dr. Holmgren, I should say, actually mentioned barcoding. So we want to know, what year was the first barcoding used in a grocery store in the US? What was the first year that barcoding was actually used for real in a grocery store, not genetic barcoding, sorry. So chat your answer in there. First answer wins a pair of socks. First correct answer. It was not 2017, but keep guessing. 75, we got a 65, 57. Man, was computers even invented yet? 79, somebody's really going for it. 88, 76, I have not seen a correct answer yet. 1980, no. 84, no. I will give you a hint. It is 1 year younger than me. I want to see what people put down at that point. 1939. No, that's not correct. Kidding. Thank you. 1950. Well, let's see. It turns out, let's see who was closest here. I think the closest I see is 1972. The actual year was 1969. So Nancy Weiner, I'm not sure how to pronounce your last name. I apologize. You have won your first pair of socks. So congratulations. Just so you know, so you can win a bar bet at some point later on. 1969, a grocery store in Ohio, a guy named Norman Joseph Woodland. So you can pull that out if you really need to. So that was the first pair of socks. Get ready for the second pair of socks coming at you. Thank you. Sorry, Weiner. So second question for you is more related to Kristen's talk directly. What is the largest crustacean in the world? The largest crustacean in the world. What is it? King crab, getting close. There you go. King crab, Japanese. I think we'll give it to Francesca Bella, and I'm sorry I don't see the rest of your name. So Francesca, you win. It is the Japanese spider crab. Well, sorry, not king crab, I guess officially. Spider crab, but I think they're different names for the same thing. It is up to 4 meter long leg span, which is just kind of creepy. So I don't believe that Kristen's Arms Hotels actually pulled any Japanese spider crabs in, but they would have to, it's just because it wasn't big enough. I think that if you had put out some bigger arms.
They're also really deep water, so they're really cool. Yeah,
that's right. Well, it would be extra creepy if they showed up in shallows in Puget Sound. So all right, so if you guys can, your winners, congratulations. If you can send me your info on how to get you your pair of socks and what size, that would be great. And yes, a UW box number is just fine for me. So Great, thank you very much. So let's get back to our speaker here. So who's got questions for our speaker? There was one earlier. Jim asked, did you measure other kinds of chemical, physical parameters, temperature, DO, pH, that kind of thing when you did your different measurements during your shoreline stuff?
Right. So we actually had an earlier study where we measured all those things at kind of a smaller set of sites. So DO, pH, and then temperature, salinity, and then a couple of lead, mostly, and like actually a whole suite of heavy metals that Jim's lab helped us with. And there weren't really any, there were some salinity effects because we had some sites like inside the Duwamish River that were really low salinity, but none of those really environmental factors had a like outsized effect. So that's why we focused on kind of high salinity areas, and that's why we measured a bunch of other things like species, algae species richness. But great question.
Cool. And did you measure those sort of once when you did your thing, so that particular day, or was it just, yeah, it's the tough thing about going out and trying to record a very changeable thing in a set location. So
yeah, I mean, that's, yeah, so for all the water quality parameters, I mean, it would be great if I could find a really nice data set that was kind of a longer time series from all those sites, but we really just measured them when we went out, although they were similar when we went out to the same site, relatively similar over and over again.
Cool. Got a question from Dr. Poole. He asks, creating a data sheet to quantitatively calculate functional diversity of the inverts is very tricky. How did you decide what traits to assess? That's
a great question. So me and Amelia read a bunch of papers on functional diversity, and we kind of like made all these lists of what people measured in other studies. And then we originally had a data set of like 20 different functional traits. And then we did a bunch of analyses to kind of figure out, like, some of them were correlated with other things. And so we just kind of chose one of those functional traits. And it turns out that some of them were not very variable at all, and some of them were very variable. And we kind of chose, I think it was about 6 or 8 functional traits that were used in a lot of previous studies. And that turned out to kind of explain the most variation in like the functional differences between communities, but it wasn't that obvious. Actually, it was a little hard to figure out.
Yeah. So people, if you have questions, please chat those in. I'll feed you one of my own. So have you try to compare your results where you actually see species that you have in your different samples, and then do genetic barcoding with something like eDNA, where you've tried to look and see whether you're getting everything, or whether that there's some chunk of of critters that you're missing because they don't like your hotels or something along those lines?
No, that would be really great. I would love to do eDNA. I don't know how to do it yet, but yep. I have a friend in Tennessee, and she does all this rare fish species detection in these rivers with eDNA. So I just got to figure out the techniques from her. I mean, that would be like a really just even like faster and less destructive method to do it. So yeah,
it's not as flashy. So sorry for folks. eDNA is environmental DNA. So it's a way to basically take, like, you can filter a whole bunch of water, or sometimes you can extract sediment and try to get sort of what DNA is out there, and then you have to be able to target the particular species. So it's a way to see what's out there, but it doesn't have the pictures that go with it, right? So you have no idea what these organisms look like, or maybe what they do to some extent as far as functional diversity goes, but you would know that you can see the ones that you do know what's there. You saw those, but then it's also, did you miss anything? It's a way to kind of look at that. So that would be a really, really interesting way to do it. So we got another question here from Leanne. She asks, How much do you think this region's diversity will help crustaceans after big heat events like the ones we had this summer, or is it too soon to tell?
I'm thinking that it's going to definitely affect the intertidal crustaceans more than the subtidal crustaceans. So like the kind of animals that I saw in my second study. I mean, I think we all heard those like reports of the like shellfish like baking in their shells during the really intense heat effect or heat wave in June. To some extent, since they can move, they can kind of like find little cracks and crevices. But it would be really interesting to tell. I mean, we're seeing a lot of shifts in kind of the kelp forest communities in Central California from a combination of the heat, which kelp doesn't like, and then the loss of like predators, like those big sea stars that we lost with sea star wasting disease. So I would love to actually go back and, you know, do the same exact study. Like this is exactly what Gary talked about in the video. You want to have some kind of a good baseline now, so you could go out in 20 years and figure out if there are changes and then which species have increased or decreased.
Cool. Got a question from Katie. She asks the invertebrates that you anticipate wouldn't use the arms substrates.
Oh yeah, bunch. I mean, so all of the really interesting kind of burrowing invertebrates, things that are going to be like burrowing in sand or mud, you're gonna totally miss those. So it's really just a snapshot of one type of diversity. And so there is going to be, you know, definitely better techniques for a lot of the kind of soft bottoms that you see in the Puget Sound. Yeah, but it does allow you to kind of look at one certain subset of animals that do like to use these and see what the diversity like is here compared to other sites.
That's cool. Actually, as a follow-up, is there, you know, the standard, the design of your arms are kind of like, they're not very fancy as hotels go. So has there been any look at like different designs allowing for different, like more different sizes and nooks and crannies that give you maybe different species?
Yeah, so sometimes people put like, you know, there's really like rough sponges that like, not like a kitchen sponge, but like a sponge that you would use for sanding. Sometimes like people put a layer of those up there. And then, I was talking to somebody at Friday Harbor who wants to put some different materials on the arms that are like even rougher to see if we can grow algae, like little seaweed on them. And so maybe, and the seaweed can maybe like take up carbon dioxide or maybe other pollutants in the water. Yeah, so you can grow you can definitely make them a lot fancier and grow a lot more things on them, depending on kind of what kind of arts and crafts you put inside the homes. Yeah.
Cool. Francesca asks, since there is more biodiversity in the northern sites, would it be safe to assume that as the larvae settle more and more inland, will they continue on this trajectory? So I think, are they more settled, such as when observing a shoreline, such as how sea stars would be closer to the water and bivalves that can hold on to water be much farther up the beach? I think it's like positioning. So is the size of like the distance that they have to travel really what's?
I feel like it's really like larvae is one like obvious thing that it seems like it should be, but the same folks who did the kind of invertebrate shoreline studies, they did a big larval recruitment study to see if larvae was the reason why there's lower diversity in the southern sites, and there was like equal larval recruitment of the little worms and clams that they were studying at both the northern and the southern sites. So that might be it, but you need more of those kind of experimental studies setting out little settling plates or whatever those larvae are going to settle out on to actually see if that's the mechanism. But
yeah, I think it would be, I don't know if you know, but there's I think the Mussel Watch, Mussel Watch nationwide. I can't remember, it's that version of it. So Washington State has done their own version because they got it, they've started to do a much more expanded version. They're doing it again this year. So they have a bunch of volunteers that set up cages at close to 100 different sites throughout Puget Sound on exactly the same week, at the same locate, the same depth of tide and everything, same mussels from the same location. So they do this whole big controlled study, which I think is really interesting. And it would be, I think, cool to see if you could somehow piggyback off of that, right? Can you use filter feeders, right, as a pass?
Well, actually, like mussels themselves, if you pull up like a chunk of mussels and like you pull them apart at Elliott Bay Marina, they have like a ton of stuff living in them. So, if you're putting out the same mussels and it's the same species and the same substrate, I mean, Mussel Watch is mostly looking at the kind of like what's accumulating in the mussels themselves, right?
Yeah.
But if you were looking at what's living on them, that would be a really cool sample.
Yeah.
That's a great idea. Yeah.
Yeah. And we can talk about it later, but they're always looking for great partnerships. And so you could have 100 different locations giving you samples all in the same way, basically using these as hotels, very porous, large cage hotels. So.
Yeah, I'm always looking for projects for marine biology. Yep. Thomas Poole, I'm looking at you. Okay. You're teaching it too, so yeah, we should talk about that. That would be really cool.
Cool, awesome. Any other questions, folks? I don't think I missed any. Oh, wait, there was a question also from Francesca. Are there veterinarians who treat crustaceans and arthropods in general? If you have a pet shrimp.
Yeah, I think like those are called like exotic vets. Like I used to have a pet axolotl, and I definitely had to bring it to like an exotic pet vet. I think it would be mostly like aquarium people. So not vets, but the people who work at like the Seattle Aquarium, or even down at Mass Marine Lab. Those are going to be the people that see the disease and kind of have a really like everyday experience with the common diseases and sicknesses. Yeah.
That's interesting because it kind of, for academia, it sort of falls through the cracks because it's invertebrates. You don't have all the you don't have a veterinarian on staff who has to keep your invertebrates alive for a study in your lab. And so
that's me.
That's right.
And I'm not a veterinarian, so yeah.
Great. So if anybody has sick crustaceans, definitely contact Kristen and she can help you out.
Yeah, no, I would like send you to other people that do this at larger scale aquariums. Yeah.
Cool. Well, thank you very much for an awesome talk. Thank you all for coming. I appreciate it. Hopefully we get to be in person sometime soon. It was wonderful to sort of see everybody. By the way, you can share if you want to turn on your camera. You guys are unfettered at this point. You can wave goodbye. Just so you know, this has been recorded. So once it's cleaned up, this will be available on the Grit City Think and Drink website. So you can, folks that that might want to see it and couldn't make it, please share that with other folks and you can see the talk. And again, hopefully see you October 12th for our next talk. Dr. Jessica Ray is going to talk. Brand new faculty up in Seattle is going to talk about her cool work that she's been doing. And then in November, we're going to hear about furniture and culture, which is a really interesting talk by Ali Madaris. And then in December, it's all about social media dating. So if you want to check that out, and I think it actually is during the time of COVID. So if you want to hear the sort of details, Ricky Thompson is definitely the person to ask questions of for our December talk. And again, if you have any ideas for speakers for 2022, please send those to me. You can email me at jimgal@uw.edu at any time. Thank you all very much. You guys have a great evening. Thank you, Kristen, for a wonderful talk. Everybody take care and be well.